Welcome you to this video lecture on glycolysis pathway and its energetics. Myself, I am Dr. N.G.P, Professor, Department of Biochemistry, Dr. N.G.P, Oxford Science College. Fate of absorbed carbohydrate. Normally, we are the carbohydrate-rich eating community people. So after a breakfast, during the digestion process, the salivary amylase enzyme which is present in our mouth, which begins our carbohydrate digestion. There is no enzyme is present in stomach to digest the carbohydrates. The next pancreatic alpha amylase further involved in the digestion of partially digested starch molecule. The next enzymes which are dextrinase, maltase, isomaltase, sucrase, lactase, enzymes which are present in this intestine which involved in the complete digestion of carbohydrates. So during digestion, the glucose molecules which enters into the glycolytic pathway. So the glycolytic pathway is describes the oxidation of glucose to pyruvate with the generation of ATP and NADH. So after the digestion process, the complex molecules are simplified into monosaccharide. The monosaccharide glucose is enters into this glycolysis pathway. In glycolytic pathway, which involved in oxidation of glucose to pyruvate, so which is get entered into the glycolytic pathway glucose, which is converted to the end product called pyruvate, thereby it generates ATP and NADH. This pathway is also called as Emmett-Mayhap pathway. Glycolysis is a universal pathway which is present in all organisms from yeast to mammals. In eukaryotes, glycolysis takes place in cytoplasm. Glycolysis is anaerobic, it does not require oxygen, but in the presence of oxygen, pyruvate is further oxidized to carbohydrate. During the absence of oxygen, pyruvate can be fermented to lactate or ethanol. The net reaction of glycolysis is the glucose along with the coenzymes NAD, ADP and along with the two inorganic phosphate which produces the products of 2 pyruvate and 2 ATP and 2 NADH, 2 water molecules. This flow chart which shows the overall glycolysis pathway. The pathway takes place with the 10th step of reaction. In the first step, there is a phosphorylation reaction where the glucose is converted to glucose 6-phosphate and the glucose 6-phosphate is isomerized to fructose 6-phosphate in the second step. In the third step of reaction, again it is phosphorylated with the help of ATP by the enzyme phosphofructokinase. Now the fructose 1,6-diphosphate is cleaved into 3 carbon sugar molecule in the fourth step of reaction. The 3 carbon sugar molecules include glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, where this dihydroxyacetone phosphate is further synthesized into glyceraldehyde 3-phosphate. So totally the two molecules of glyceraldehyde 3-phosphate is synthesized after the fifth step of reaction, which is further dehydrated to 1,3-bisphosphoglycerate and further it is dephosphorylated to 3-phosphoglycerate. Again it is mutated to 2-phosphoglycerate. Again there is a removal of water molecule in the ninth step of reaction which is synthesized into phosphoenol pyruvate. Again in the 10th step there is a generation of ATP molecule with the synthesis of end product called pyruvate. Overview of glycolysis. The glycolysis is divided into two pathways or two phases. In the phase 1, up to the 6-carbon glucose to 2-carbon sugar conversion, the energy is invested. But in the second phase of reaction, the energy is synthesized. We are going to learn individual steps of reaction in glycolysis from the following slides. In the reaction 1 is phosphorylation. The mechanism of reaction is phosphorylation, where the glucose is phosphorylated to glucose 6-phosphate. This glycolysis takes place in the cytoplasm of cells, and glucose enters the glycolysis pathway by the conversion to glucose 6-phosphate. Initially, there is energy input corresponding to cleavage of inorganic bond phosphate bond of ATP. The enzyme which involved in the catalysis process is kinase or hexokinase, which catalyzes the phosphorylation of a molecule using ATP. In the reaction 2, which is about the isomerization mechanism, where the isomerase enzyme catalyzes the transformation of compounds into their positional isomers. In this case of sugars, this usually involves the interconversion of aldose into ketose sugar or vice versa. So there is the glucose 6-phosphate is isomer of fructose 6-phosphate, where the free energy synthesized in the step is 1.7 kilojoule per mole. And the reaction 3 explains the mechanism of phosphorylation, where the glucose fructose 6-phosphate is again phosphorylated in the presence of ATP to fructose 1,6-bisphosphate. The transfer of phosphoryl group from ATP to carbon 1 of fructose 6-phosphate to produce fructose 1,6-bisphosphate. So this step is important, irreversible and regulatory step. The enzyme phosphofructokinase is one of the most complex regulated enzymes with various allosteric inhibitor and activator. ATP is an allosteric inhibitor and fructose 1,6-bisphosphate is an activator of this enzyme. ADP and AMP also activates phosphofructokinase, whereas citrate is an inhibitor of this enzyme. The reaction 4 of this glycolysis cycle, where the fructose 1,6-bisphosphate 6-carbon sugar is cleaved into two 3-carbon sugar that are dihydroxyacetone phosphate and glyceroldigate 3-phosphate. Again, in this step, dihydroxyacetone phosphate again it is converted to glyceroldigate 3-phosphate by the enzyme triose phosphate isomerase. Aldolase enzyme catalyzes this conversion of fructose 1,6-bisphosphate to dihydroxyacetone phosphate and glycerolidate 3-phosphate. And the reaction 5, that's isomerization, is a mechanism of reaction where in this fifth reaction, again the dihydroxyacetone phosphate is catalyzed to the product glycerolidate 3-phosphate. Reaction 6 is an oxidation reaction where the glycerolidate 3-phosphate along with one inorganic phosphate and the coenzyme NAD, which is synthesized into 1,3-bisphosphoglycerate, where in this step glycerolidate 3-phosphodehydrogenase enzyme utilizes the NAD as a coenzyme along with the liberation of 2 hydrogen from this first carbon of glycerolidate 3-phosphate, where there is attachment of phosphate molecule, thereby it synthesizes 1,3-bisphosphoglycerate. Reaction 7, the substrate level phosphorylation occurs where the enzyme phosphoglycerate kinase catalyzes the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate along with one ATP molecule is synthesized. Here you can see this is the ADP, adenosine diphosphate. Along with that, after the synthesis of 3-bisphosphate, one phosphate molecule is pink color. This one phosphate molecule is utilized by the ADP. Thereby it synthesizes one energy molecule ATP in this step. Reaction 8, it is a shift of phosphorate group. So 3-phosphoglycerate is shifted to 2-phosphoglycerate. The enzyme catalyzes this reaction is phosphoglycerate mutase. The mutase is an enzyme that catalyzes the transposition of functional groups such as phosphatases, sulfatases, etc. Here it transpositions the phosphate group. The reaction 8, there is an active site of histidine side chain participates in the phosphate transfer by donating and accepting a phosphate. So the detailed explanation is given in this step. This is the enzyme structure where the histidine is an active site. Where it binds with the substrate, it also participates in phosphate transfer by donating and accepting the phosphate. This process involves 2,3-bis phosphoglycerate as intermediate. So there is one more intermediate is synthesized in this step. Phosphoglycerate mutate reaction, there is a conversion of 3-phosphoglycerate to 2-phosphoglycerate. In active form, the phosphoglycerate mutase is phosphorylated at histidine 179th position. There is a transfer of phosphoride group from enzyme to 3-phosphoglycerate, generating enzyme-bound 2,3-bisphosphoglycerate intermediate. This compound has been observed occasionally in the reaction mixture. In the last step of reaction, the phosphoryl group from the carbon 3 of intermediate is transferred to the enzyme. 2-phosphoglycerate is released. So in most cells, 2,3-bisphosphoglycerate is present in very trace amount, but in erythrocyte it is present in significant amount. There it regulates oxygen affinity to hemoglobin. The reaction 9, there is a dehydration reaction. 2-phosphoglycerate is dehydrated to phosphoenol pyruvate, which is the enzyme enolase, which is a magnesium dependent enzyme. The last step of reaction is also substrate level phosphorylation. Here also there is a generation of one more ATP molecules. The end product pyruvate is synthesized in this step with the help of enzyme pyruvate kinase along with its cofactor magnesium. What is the energetics of glycolysis pathway? Where the ATP is formed in 3-step of reaction and the ATP is consumed in 2-step of reaction. Totally the ATP is synthesized, number of ATP synthesized is 8 ATP molecule. Thank you for patient listening.